Electrophoresis. If we dissect the word itself, we'll get the terms electro and phoresis. The suffix phoresis means movement or transmission, while the prefix tells us that this process involves electricity. And in simple terms, this is exactly what electrophoresis is: movement that is induced by electricity. This laboratory technique is commonly used by biochemists to separate segments of charged molecules such as DNA, proteins, or RNA. So why do we even use DNA or proteins or RNA? Well, from our biology background, we know that DNA has three major components that make up its entire structure: the nitrogenous base pairs, adenine, thymine, cytosine, and guanine, and they're neutrally charged; the sugar backbone, also neutral; and the key component that makes this entire process possible, the phosphate groups on the backbone of the DNA structure. And basically, when you turn on the voltmeter, the electrical current will flow into the apparatus, and since DNA is negatively charged, we're going to place it at the negatively charged end, and that will allow the DNA to migrate towards the positive end. So there are two procedures within this experiment that you have to complete in order to begin the lab. So one, you have to prepare the DNA sample and extract the DNA, and two, you have to prepare the gel. So to extract the DNA and quickly explain how it's done, you would first extract the cells by either taking a cotton swab and then rubbing it on the inside of your mouth, and then you would have to place them into a salt solution so that the positively charged sodium molecules can protect the negatively charged phosphate groups on the DNA. 2. You have to add detergent to break down and remove the sample of any phospholipid bilayers in order to lyse the cells. Basically, you're inducing cell lysis. And then you'd add protease, which is an enzyme, into the sample to break down any proteins, because you want to purify the sample as much as possible. So now that the entire beaker or sample that you have is mainly DNA, you want to precipitate the DNA. When you introduce DNA with ethanol or isopropanol and salt, the DNA becomes insoluble in the water, and you'll see it start to precipitate, and you'll actually be able to see like a string-like structure. After that, you take a sterile pipette and then you just slowly take it out of the beaker, and then put it into another beaker containing an alkaline buffer solution in order to further purify it. So now that you've extracted the DNA, you want to start preparing it for the lab. Basically, what you're going to do is introduce restriction enzymes into the test tubes. Restriction enzymes are like scissors that cut the DNA into whoever knows base pairs. By introducing different types of restriction enzymes into the test tube solution, the DNA you previously left in the test tube will begin to break down into DNA segments of different lengths. Once the reaction is complete, simply add a bit of dye into the DNA samples and you're ready. So while the restriction enzymes are doing their job, it is best to begin preparing the agarose gel, which is responsible for acting as a medium in which the DNA fragments will be electrophoresed upon turning on the power supply. Agarose is insoluble in room temperature, however when mixed with a buffer solution or boiling water, it will dissolve and cool down to the gel-like substance as it molds into the shape of the electrophoresis apparatus. Once the DNA sample is added to the wells, the power supply is turned on, and the DNA sample will move towards the anode due to the negatively charged phosphate groups on the DNA's backbone. So if you were to shrink down to the size of the DNA fragments, you'll see that the actual structure of agarose resembles a Rice Krispie or a cancellous bone tissue due to the porous structure. This is good because this type of structure allows DNA to move through, and the smaller pieces of DNA will move through the matrix fairly quickly relative to the larger chunks of DNA that take much longer as they get tangled. So after a certain amount of time, you will see these bands form, and each band represents a certain length of DNA segment. If we think of the entire lab as a race, and all the DNA segments are racing towards the finish line, it's as if the anode is the finish line, passing through all these obstacles in the agarose solution. And those that are smallest can easily fit through these obstacles, which is why they are in the lead, while the larger ones are far behind, as they have a much tougher time passing through the obstacles. Now, to study your results, you can just peel off the gel and examine the band. Some real-world applications of electrophoresis include paternity tests, forensic analysis, and DNA analysis.